Hello, sir. Hello.
Where we left yesterday. Okay. Yesterday we finished one question. This one from the chromosome. No, that this question number 104 that has been given. Mm, 104. This question, I think we saw here. This diagrams were there, and this shape of the chromosome. Okay, so all these metacentric, submetacentric, ectocentric, telocentric chromosome we have seen. So this is the shape of the chromosome based on the position of centromere in the chromosome. And now next question is there that here is which of the following statement is correct, is not correct. Okay, now which of the following statement is not correct? Statement A single human cell has 2-meter long uh, thread of DNA. Is it the correct thing? The DNA is in our body that is very long. Actually, we see within the microscope that when we see during the cell division, at that time the DNA is actually very much condensed condition, and this condensed condition, that's why we see the length of the chromosome only few micrometer in length. Hmm. So this statement, the one, a single human cell has 2-meter long thread of DNA. Part of the chromosome after second constriction is called telomere. This is one statement. Another, the centromere forms the first site of constriction. The chromatin consists of DNA, RNA, histone, and the non-histone protein. Okay, this so which of these statements are correct? Or most of the statements, all these out of here, you will get 3 statements are correct and 1 will be wrong. So what is that? Which one? Can you tell? See, this one I told that it is true. This one is correct statement. This DNA, which usually we see in this way, is it that whenever we talk about the structure of the DNA, then this is the structure of the DNA we see. This is a long thread made of different types of nucleotide. DNA is a polynucleotide chain, is it? It is a polynucleotide nucleotide chain. This is a polymer of deoxyribonucleotide. And this DNA this DNA is again is associated with the protein. So that histone proteins are there, and this histone protein, this DNA is this DNA wrap around this protein molecule. So due to this, again also the length of the total length is decreased because it is coiled around the protein molecules. This forms the structure that is called the chromatin. So chromatin is DNA and protein. Then this one is in the interphase nucleus. When the cell is in interphase, during this, the DNA portion that remain in the form of chromatin thread. Okay, and this is also very long thread, tangled mass of thread seems to be. So this is chromatin. But at the time of cell division, when the cell division progress in mitosis or meiosis, this thread, this chromatin thread, again it condensed, it coiled. This chromatin thread is there. This chromatin thread is again coiled. This is the formation of this we call the solenoid structure of the chromosome. Here is the DNA. This one will coil around again, coil. So this way, and after this will again further coil or it will fold, and that's why this become very short and very thick, so we can see them. Otherwise the DNA is, diameter of the DNA is, do you remember that only 20 angstrom is the diameter of the DNA, right? So this become folded, and that's why only in it is in micron, but total length is very long. So if all our 46 chromosome in a cell, how many chromosome? 46 number chromosome. If their DNA is stretched, then they will be nearly 2 meter long thread of DNA. This part of the chromosomes after the second constriction. Say, if we see the structure of the DNA, see here, I will draw the structure of the DNA. Okay, this is, and this one. here little bit correction. Okay, now we see here this is the place. Here it is one constriction. This constricted area, this is known as the primary constriction in the chromosome. So chromosome, this one is a chromosome that we study at which stage? That is a metaphase of mitosis is the best stage to study the chromosome structure and its morphology. Okay, so this chromosome has two chromatids. These are the two chromatids. And these chromatids are attached at one point. This is the place where the two chromatids are attached. This place is known as centromere. Okay, and the centromere is located at primary constriction. All right, and the chromatids on either side of the constriction, primary constriction, these are called, that is the arms of the chromosome. So it is one arm of chromosome. So here in this chromosome, this one is shorter, this one is longer. So it is a long arm and short arm. So arm of the chromosome. And this is the primary constriction. This primary constriction is present in all the chromosome. This is the end of the chromosome. Okay, this is end of the chromosome, and end of the chromosome is known as telomere. Telomere, that is the end, and telomere has a very characteristic feature that these telomeres are non-sticky. And due to this non-sticky nature, non-sticky nature, the one chromosome, they do not get attached with other chromosome. So the individuality of the chromosome is maintained within that small space of cell and the nucleus. Cell is microscopic. Within that nucleus is further smaller structure, but within that, so the 46 chromosome, and even in some of the organism there are 1,000. In some plants it is more than 1,000 chromosome per cell, but all the chromosome maintain their individuality, and this is due to this non-sticky nature of the telomeres. Okay, if this telomere portion break, then what will happen? Either these two chromatids will get attached to each other, or one chromosome will get attached to the another chromosome. हेलो, थोड़ा बाद में फोन करो. हाँ. So this This is the characteristics which are of telomere. So all the both the end there is telomeres. And here in some of the chromosome there is another constricted region. Okay. See here I have shown another constricted region, but this constricted region is not present in all the chromosome, in some only. So this one is a additional constriction in one of the arm. Usually it is in the long arm. Okay, and this one is known as secondary constriction. Okay, this secondary constriction actually this is the site where the nucleolus is organized. Okay, secondary constriction is the site where nucleolus during interphase we can see within the chromosome there is a nucleolus. So secondary constriction is the site where nucleolus is nucleolus is organized around this portion of the DNA. At this portion, actually nucleolus, these are the site where the RNA and the proteins, they are get associated to form ribosomal subunit. Ribosomal subunits are synthesized here. Here the RNA, ribosomal RNA is synthesized and proteins comes, and with that ribosomal subunits are synthesized. And that's why it makes a ball-like structure, that is we call the nucleolus. And due to this secondary constriction is also known as the nucleolar organizing region. This is also known as nucleolus organizing region. Okay, so as nucleolus organizing region. So it is NOR. In brief, we call NOR. And this chromosome, which is having the secondary constriction and the nucleolus organized region, they are called nucleolus organizing chromosome. Nucleolus organizing chromosome. So that is NOC. It is nucleolus organizing chromosome. Okay. So this nucleolar organizing chromosome, this gave the chromosome a definite shape, and so they are useful tools, morphology for the identification of particular chromosome. And the portion of the chromatid which is beyond this secondary constriction, usually they are very small and form as a small ball-like structure. All right, a small ball-like structure, and this small portion of the chromatid, this one is called satellite. Okay, so this is satellite is there, it is attached, it is satellite is present at the distal end of the chromosome which have the secondary constriction. So this is,
Excuse me, sir.
yes,
What is the function of the satellite?
no, this is they also contain some of the genes. Okay, there is not a particular things, but this is the portion seems to be hanging just somehow thinly attached with the main body of the chromosome, so they are called the satellite. Okay, but it is function is here in the secondary constriction. This may contain some of the genes, so as usual, as it is made of DNA, so genes are there. Special things are not there, and it, of course, it help in the identification of the chromosome. Okay, so different chromosomes are identified according to their length, position of the centromere, presence or absence of secondary constriction, and certain other feature. Okay, so this is satellite. So this satellite containing chromosome is also known as SAT chromosome. Okay, they are also called SAT chromosome. Satellite. So this is the all about the structure of the chromosome. And here in the centromere, there is another structure is found, that is what we call the kinetochore. Kinetochore. This kinetochore is the structure that which the sticky substance or the site to which the spindle fibers are attached. Okay. So kinetochore is the structure which is present in the centromere, centromeric region, and to that spindle fibers are attached during the cell division and the chromosomal movement during cell division. Okay, now let us come to the main thing, that is the question we were discussing. Huh? That so the part of the here, the first one we have, that I told that it is correct. And the second one, the part of the chromosome after the second constriction is called telomere. No, telomere is that part of the chromosome after constriction. This is actual is that should be satellite. Is it? Satellite will be the correct word here. And this one, so part of the chromosome after the second constriction is satellite. So this one, this statement is wrong. Centromere forms first site of construction. That also I told, that centromere is present where? Centromere is present in the primary constriction region. Is it? It is in the primary constriction region. Then, so this one forms the first constriction, and the chromatin consists of DNA, RNA, histone, and non-histone protein. See, main component of the chromatin, main component of the chromatin is DNA plus histone protein. DNA and histone, this one forms the chromatin. But beside this, when the chromatin is organized into a further condensation takes place, coiling takes place, during that case, there is some non-histone protein also. Okay, non-histone protein also, and some type of a small RNA, that RNA which are very small. Okay, this is sometime called the small chromosomal RNA. The RNA which is required for the organization and stabilization of the chromosome, they are known as chromosomal RNA. They are even smaller than the tRNA. You know that we usually call that the tRNA is the smallest. No, there are, we usually know there are three type of RNA. One is the ribosomal RNA, messenger RNA, and transfer RNA, tRNA. And tRNA is the smallest. But no, the tRNA is the smallest among those three. But there are some smaller RNA also is there to stabilize the structure of the chromosome, this chromosomal RNA. This is a smaller chromosomal RNA. So this in this statement, this one also correct. So they contain DNA, RNA, histone, and non-histone. This is also correct. This is also correct. Here is the second one is not correct. So our answer is second one. Okay. Come to next one here. This one I think you can easily solve. Mm, Here is the question. The
Option B, sir.
uh,
It's
option
option B. Option B, sir.
satellite B is secondary constriction, and C the diagram C is the type of chromosome. Type of chromosome. This one is almost metacentric. No, so it is metacentric. Centromere is in the middle. So our answer is B. So this was based on that. Now come to some of the question based on this uh, based on the some statement, correct or wrong. This is I think it will be clear for you to see. Let's say this question. Mitochondria and chloroplast are the two statements are there. One is semi-autonomous organelle and formed number two statement is formed by division of the pre-existing organelles, and they contain DNA but lack protein synthesizing machinery. Which one of the following option is correct? This question came in 2016 NEET exam. And what is this? Two 2 is true, but 1 is false. 1 is true, but 2 is false. Both 1, 2 are false. Both 1 and 2 are correct. This is so which one do you think that what should be the answer? What should be the answer? Any idea? Is what about this statement, the semi-autonomous organelles? Mitochondria and chloroplast are semi-autonomous organelles. Actually, autonomous is one who can run themselves. They can carry out their all the needs. Is it? They can multiply, they can grow whatever the enzymes, etc., proteins and other things that is required, they can synthesize, then it is autonomous. Okay, but here it is said it is semi-autonomous. This statement is true. Mitochondria and chloroplast, these two are the semi-autonomous organelles. This is true, but what about this two? Number two, that formed by the division of the pre-existing organelles. Say, actually, within the cell, the number of mitochondria, number of chloroplast, this plastids, their number also multiply. Okay, they are not just synthesized. Say if there is one mitochondria, this mitochondria may break and this will form two mitochondria. Okay, this takes place by fission also, that yesterday we have discussed this one. Uh, Chloroplastid also they divide. Okay, during the cell division and the cell growth, when the cell is preparing for the division, then this mitochondria and chloroplasts they divide, and almost nearly half of goes to the two daughter cells. Of course, the young cells, when it is growing and according to the need of the cell, according to the metabolic activity, this uh, chromosome, the mitochondria, their number also will increase by the multiplication. This is true, formed by the division of the pre-existing organelles. All right, which one is already present from the beginning. So by the division, and they contain DNA. This is true, but lack protein synthesizing machinery. DNA, if it is the mitochondria, okay, in the mitochondria there are some DNA is there, and this DNA is the circular DNA like that of the prokaryote. They also contain RNA, okay, and they also contain the ribosome. They contain DNA, they contain RNA, they contain ribosome, and the enzymes required for the synthesis of protein. Enzyme for protein synthesis. Okay. So machinery for protein synthesis is there. Machinery for the protein synthesis are RNA. All types of RNA are required. mRNA is required to carry the message from the DNA. They require the tRNA, okay, and they require amino acid. They require some enzyme. All these things and ribosome, all these things are required for the protein synthesis. So protein synthesis machinery is also present in mitochondria and plastid, and that's why they can synthesize some of their needed protein and enzyme, some metabolic activity they can control themselves, and that's why they are called semi-autonomous. Okay, they can call semi-autonomous. So this statement that but lack protein synthesizing machinery, this one is not correct. These things happen, this type of system is present in chloroplast also. So chloroplast also has some. And as they have DNA, so naturally they contain some genes, and they contain some gene, so they can influence the inheritance. They also carry certain, or they can inherit some of the parental character to the next generation. That is what we call extra nuclear inheritance. Extra nuclear inheritance. Okay. Or sometimes called the cytoplasmic inheritance. Okay. So this is, so here our answer will be 1 is true but 2 is false. This is, first statement is true, second statement is partially false. Up to here is true and this portion is false. All right. Next question. Study the following statement regarding the mitochondria and select the correct one. These are the site of aerobic respiration. Matrix contains single circular double-stranded DNA molecule, a few RNA molecule, and 7ts ribosome. Mitochondria divide by fission. Mitochondria are fully autonomous. So, which of these are correct? And select the correct one. Correct one is, see, this is another type of how many correct statement. One thing is that how many correct statement, and another is which one is correct, which one is wrong. So which statement is correct here? What about the first one? First one is correct?
Yes, sir.
This is correct statement. Second one, that the matrix contains single circular, this is a DS is double stranded. Okay, DNA, that is a double stranded molecule, say few RNA and 7ts ribosome. Say organelles, ribosomes are prokaryotic type. Remember, in this eukaryotic cell cytoplasm, the ribosomes are 80s, but those ribosome which is present in mitochondria and chloroplast, they are not 80s. They are prokaryotic type 70s. In mitochondria, it is 70s. In chloroplast, it is little bit less or more, not exactly 70s. Okay, but it is not 80s. So it is prokaryotic type. 70s. This statement also is correct. Mitochondria divide by fission. This is also correct. I have already discussed that they divide, and mitochondria are fully autonomous. Is it fully autonomous? No, they are semi autonomous. That is, complete autonomy is not there. They are semi autonomous, not fully autonomous. So, number four statement is wrong. So, our correct statements are 1, 2, and 3. So, C is the answer. Next question, say more or less similar type of that three four statements are there, and you have to select the correct or wrong answer. Consider the following statement and choose the correct option. The endomembrane system include the plasma membrane, the Golgi complex, lysosome, vacuoles, all in the endomembrane system. This true. All the membrane and tubular structure, they are included in the endomembrane system. Okay. Endomembrane system includes the plasma membrane, all this endoplasmic reticulum, Golgi complex, lysosome, vacuoles, all these are endomembrane system. ER help in the transport of substances, synthesis of protein, lipoprotein, and glycogen. Lipoprotein synthesis takes place in the Golgi complex. Okay. This ER in the mainly it is synthesis of proteins. Ribosomes are involved in protein synthesis. That is true. Mitochondria help in oxidative phosphorylation and generation of ATP. This is also true. So this one true, and this one true. This two is lipoprotein. Only one. Substances, and choose the correct option. Choose the correct option. So this is true. This one is all lipoproteins. Lipoproteins, it is a lipid protein. This synthesis of lipoprotein is the function of mainly the Golgi complex.
Excuse me, sir.
Hmm?
Excuse me, sir.
Yes.
Is this lipoproteins is lipids?
Lipoprotein means lipo is, that is for the lipid. Okay. And associated with protein means lipid containing protein, or you can say protein containing lipid. Means two are there. So it is lipo. One is called glycoprotein. Lipoprotein, glycoprotein. Glycoprotein is, glyco is carbohydrate, sugar. Glycogen, okay. So this is sugar, sugar containing protein. These are the compound, okay, compound protein or the compound lipid. We have so glycoprotein, glycolipid, this type of word content. Lipoprotein, lipid containing or protein containing lipid. Ribosomes are involved in protein synthesis. This is true. This one is true. So this is this one is confirmed correct, that I will say. And there is nothing like option. No, one, three, four option is not there. It means this one is a lipoprotein. Some of the lipoproteins are also synthesized in the plasma.
Excuse me, sir.
Yes.
The smooth endoplasmic reticulum also synthesis lipids, right?
Yeah, that is a smooth one is for the synthesis of lipid. And here what happened, that lipoprotein means, say, lipid has been synthesized separately, protein has been synthesized separately. And after that, this lipid and protein are joined. That forms the lipoprotein. Okay, clear?
Yes, sir. Thank you.
These are actually synthesized separately, and then some of the portion they are joined. So here our answer will be A. All right, 1, 2, 3, 4. But the main portion of the lipoprotein synthesis, and that is the organization, that was the um, what is Golgi complex. Yesterday we did know this question, the function of the Golgi complex. I did one question. Yes, the important site of formation of glycoprotein and glycolipids. This Golgi complex. But means that endoplasmic reticulum also, they also take the okay. Now come to some of the question based on the matching. Okay. See here, this is, there is only by no reasoning, nothing, that here is simply column 1 and column 2 we have to uh, match. Here is on one side the name of the scientist, and this side is a related discovery in the column 2. Here it is a Leeuwenhoek, Robert Brown, Slyden and Swan. So what is this side? First saw and described living cell. Presence of cell wall is unique to plant cell. This was a discovered nucleus. All plants are composed of different kind of cells. Say, Leeuwenhoek, this is, it is the first time that 1 to 1. Don't get confused. Sometimes they give correct also, correct same matching on the same line. Okay, so Leeuwenhoek first saw and described the living cell. Robert Brown, he Robert Brown was with the discovery of nucleus. Sliden, a botanist, he was all plants composed of different kinds of cells. C is 4. And Swan was a zoologist. He studied plant cell as well as animal cell. And Swan told that the cells are bounded by plasma membrane, that a limiting membrane, cell membrane is there. Okay, but in plant cell, there is cell wall, and that is a external cell. This was told by Swan. He studied plant cell as well as animal cell. A with 1 is this one and this one, and also this one. Oh, so this cannot be correct. Now you go to B. B is 3. Is it? This is this here. B is 3. So now your competition is the selection is between A and B. D will you can discard. Okay. Number C is 4. C is 4. Which one? Here C is 2. C is 4. So your answer is this one. No need to check the D, because from here after that there is nothing. Okay. Next is um, say this question. This is also matching, and in this matching your okay, RBC, WBC, columnar epithelial cells, and nerve cell. This is the type of cell, shape of the cells it has been given. And in the this side, name of the different cells, and this side their shape. Okay. So in the column 2 is their shape. So what is this? Which one? RBCs. RBCs are what type of cell? Branch and long. Which cell is branched and long?
Nerve cells.
Yes, nerve cells have a branch and long. WBC is amoeboid. Amoeboid, you understand? You know amoeba?
Yes, sir.
Amoeba, unicellular animal, aquatic animals, and the amoeba shape of the cell is very irregular and it keep on changing. Okay, they have the pseudopodia, no? These things keep on continuously changing. So it is a irregular shape. All right. So that's why it is amoeboid, means it is amoeba-like, more or less, that which keep on changing of different shape, not perfectly that is spherical or oval or it is. So this is amoeboid. Columnar epithelium. It is columnar. Column means pillar like. Epithelial cell. Epithelial cell is found in the surface of the organ. Okay. So epithelial cells is epithelial cells in case of animal body. This is not in plant. All the of course all these cell types they are in the animal cells. Hmm. This epithelial cells they are see our intestine is there, alimentary canal. So that inner layer of this alimentary canals or the other organ, they are lined with the epithelial cells. Okay, the epithelial cells are this one will be
Option 4, sir.
option 4, long and narrow. Okay, and RBC is round and biconvex. Actually, RBC, if you see from the top, no, they are circular, disc-like. But what happened? Their middle portion is pressed a little bit less thicker. Margins are thicker, and the gradually the middle portions are the middle portion the thickness is less, so it is a concave and it is a biconcave. So from the side if we see by sectioning, then it will appear like this, that the middle part is thin and the margins are thicker, so slightly biconcave. So our answer will be A with 2. A with 2 is, see here, this one is very, very easy, that the options given. If you know that A is with 2, confirmed, then you need not to check others, because in no other option A has been given with 2. In the B, A is 3, D, A is 4, C, A is 4. So all these options cannot be. So if you see only this one, then you just put the correct sign here. Don't go to match with other and spend your time. Okay. Clear? So this way also during your exam, you can save your time. Okay. Now come to slide. Here is matching. Just quickly we will, it is time. Quickly we will see endoplasmic reticulum. Here is cell organelle, this side is function. Endoplasmic reticulum, free ribosomes, mitochondria, and contractile vacuoles. Okay, endoplasmic reticulum take part in cellular respiration? No. Mitochondria take part in cellular respiration. Then take part in osmoregulation. And excretion, this is this is carried out by contractile vacuoles. No, contractile vacuoles are in some of the animal cell. Okay, the vacuoles are there. Particularly say that in amoeba, I told that amoeba they have food vacuole and contractile vacuoles. Okay, this contractile vacuoles and contractile vacuole particularly maintain the osmoregulation, means they are maintaining the osmotic pressure within the cell. See, this if they do not maintain the osmotic pressure of the cell, then what will happen? If the osmotic pressure is high, the cell will absorb water and the cell volume will keep on increasing and ultimately it will burst. So they have to maintain the osmotic pressure in such a way that not much of the water is move inside the cell. So when this water is moved inside this contractile vacuole, they increase in their volume and come to the margin, and after that it bursts and releasing all the water outside of the body. And along with that, some of the excretory product also is sent out from the body. So this contractile vacuole help this unicellular organism to maintain their body shape, size, and for the excretion. Next is free ribosome, synthesis of lipid and endoplasmic reticulum. Endoplasmic reticulum is synthesis of lipid. Endoplasmic reticulum, that is the smooth one. This is the lipid synthesis. And free ribosome, synthesis of non-secretory proteins. That is the those which are not attached to with the endoplasmic reticulum. Actually, secretory one means it has to be transported outside to somewhere. So, endoplasmic reticulum, actually, this is a channel, this is tube for the transport. If the protein has to be transported, so the ribosomes, they should be very near to the endoplasmic reticulum, so that as the protein is synthesized, this protein move into the lumen of the endoplasmic reticulum and it is transported. So those which are not required to transport, to be used locally, they are synthesized by the free ribosome in the cytoplasm. So our matching answer will be A with 3. So A with 3 is, see here, all these A, C and D is A with 3. So A, C, D. Now second one, B with 4. B with 4, which one? Only A. So A is our answer. Don't go to C anymore, because nowhere B is with 4 except that A. Okay. So here is our answer. Next is, okay, this it is time. Hmm. Next one, okay, let's see quickly. Next one, with this I will end. Match the following. Here the cell organelles, lysosome, ribosome, smooth endoplasmic reticulum, centriole. So you will prepare that all the cell organelles and their function, cell organelles and their function, that should be, you can prepare a list. uh, you can, in your note you can make one place where in one side you write all these cell organelles, and other side against it you write the correct function that it carries. So if you do that, means you may get one or two questions from there. Lysosome, ribosome, smooth endoplasmic reticulum, and centriole. So lysosome is, they contain hydrolytic enzymes. Digestive enzymes are there. These hydrolytic enzymes break down the large molecule into a smaller one. Means partial again digestion. Okay. So these are the lysosome. They break down the large molecules, protein, the large carbohydrate, large fat, okay, are broken down by the lysozyme. Ribosomes. Ribosomes, protein synthesis. Smooth endoplasmic reticulum. Smooth endoplasmic reticulum means they are not for the protein, they are the steroid synthesis. Okay, steroid, that steroid and fat, lipid. Okay, so they are actually steroids, so they are actually the lipid by nature. So this is C is this one. Centriole is for spindle formation. So this C to 3, D to 4 means giving two, only two are C to 3, D to 4 is which one? C3, D4 is this one. Okay. That is, our option is this. Correct option here. Also the next one, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, lysosome. This one, intracellular, extracellular digestion. Intracellular and extracellular digestion. Just now I told, this is the lysosome which contain the digestive hydrolytic enzymes. So breaking down of large molecule into a smaller one. So this is intra and extracellular digestion. Golgi complex is moves material out of the cell. Lipid synthesis. ACR is lipid synthesis. This is RER, rough protein synthesis and secretion. Golgi complex moves material out of the cell. Actually, Golgi complex, their function is packaging. They are the industries, that is the packaging industry, the final product, finishing product they gives, Golgi complex. So material which are synthesized, they are packed, and then the secretory vesicles, etc. are there, they are sent out of the cell, most of the cases. So the answer will be 1, 2, 3. One is A to 3 and B to 2. C is 4. D is 1. Yes, this one is the answer. Okay, so this is today. We will end here. You can practice. Questions are many. Of course, if you see here, there are several questions has been given. If you can, there are many questions that was asked in the NEET in different years. So these are the very important questions that you can practice. Okay. In next class will be for the biomolecules.